Thanks, Scott. That was great. Now, do we have Ciemon Caballes? Excellent. He's going to discuss insights into the reproductive biology of Cots and management.
Ciemon, sorry, just before you start, I just wanted to make a quick announcement to the group. We have quite a few questions coming in for this session, and one of the features of Slido is that you can vote for a question that you would prefer to be answered in person. We're not going to be able to get through all the questions, so I ask if you do have a quick moment throughout these last presentations just to highlight some of the ones you think would be better to answer, and then we'll just go to the top 4 or 5 at the end of the panel. Thanks, Ciemon.
Thank you. Good morning, everyone. First of all, I'd like to acknowledge the traditional custodians of the land where we do our work, and also recognize their continuing connection to land, sea, and community. My talk this morning is on the reproductive biology of Cots and what are the implications of this on managing population outbreaks. I've worked with Cots, I think, over the past 15 years and studied their sex life over the past 5 years. So it's become an obsession at this point. And yeah, I still have 4 Cots spines lodged in my knee, so I can say that I'm part crown of thorn starfish as well. So the effects of Cots outbreaks, the impact of Cots outbreaks have been in most places around the world have been far greater than other disturbances. For example, this in Moorea in French Polynesia, Cots outbreaks have resulted in a over 90% mortality of corals, and the geometric rate of change in the coral cover due to Cots was reductions in coral cover were more compared to cyclones, bleaching, and other multiple disturbances. Here in the Great Barrier Reef, Cots have also caused, so this is a study before the mass bleaching events, but Cots have resulted in loss of coral cover more than cyclones, bleaching, disease, and other multiple disturbances. And the time to recovery for Cots outbreaks as well is longer compared to bleaching, disease, and storms. Again, this was before the successive mass bleaching events from 2016. So you can see that Cots outbreaks take from 6 to almost 9 years to recover. So why do we manage Cots? So Cots predation appears to be the most amenable to intervention. And also with the additional threat of climate change, the impacts of climate change, mass bleaching events, it provides renewed incentive for us to manage and mitigate all sources of coral mortality. But the long-term and permanent solutions require an improved understanding of the ultimate causes of these outbreaks. So why do outbreaks occur? So this debate is still continuing, but as Dan showed, populations are spatially and temporally patchy, and this points to major fluctuations in reproductive success in Cots. So addressing these crucial knowledge gaps in the reproductive biology, ecology, and early life history of Cots is key to identifying this population bottlenecks and understanding what's ultimately causing these population outbreaks. So this is the Cots life cycle. So it goes through fertilization and embryogenesis, and then the larval stage, which Maria showed us, and also settlement and recruitment stage. And for this talk, I'll be focusing on adult reproduction, ecology, and spawning in adults. So you might think that we've studied we've already studied Cots to death, but there's still plenty of knowledge gaps in terms of its biology and ecology that would help us identify this key bottlenecks. So for this talk, I'll be looking at these key knowledge gaps: sex ratios in the field, their size at reproductive maturity, their fecundity, and their spawning behavior. So these are from samples from 2013 to 2014. So this was sampled prior to the spawning season, so between September and November. And it shows us that the sex ratio is slightly skewed towards males in all but 2 populations here. And although only 5 out of the 13 reefs sampled were significantly different from the expected 1-to-1 ratio. So this may reflect male-biased populations, could be an adaptive strategy to offset sperm limitation. So this study by Russ Babcock shows that fertilization rates are always higher for if there are more males. So this could also be pointing to sperm limitation, but also increased incidence of polyspermy. Size at sexual maturity. So the proportion of sexually mature starfish were also collected prior to spawning, and we fitted it in a logistic curve. And this, so the size at onset of maturity, 50% of Cots were reproductively mature at around 173 millimeters and at a weight of 203 grams. And at a diameter of around 208 millimeters, 90% of the Cots were reproductively mature, and also at a weight of 322 grams. So if we use derived growth curves from previous studies, Lucas, and from lab studies and field studies by Richard Stump, we could estimate that size at age at sexual maturity is around just before 2 years. So 50% of them are reproductively mature just before 2 years, and 90% of them are reproductively mature around just 2 years. So the onset of maturation is related to starfish size and age. And also in the field, it's been observed that the largest individuals are usually the ones that first exhibit gametogenesis in any given cohort. So we measured reproductive allocation in Cots. So that was from a pretty large starfish, almost 1 kilogram of gonads. And so for female and male Cots, reproductive allocation in females based on the weight of gonads is higher than in females compared to males. So for a 40 centimeter starfish, it's 20% of its weight is made up of gonads. And for males, same size starfish, around 10% of its weight is made up of gonads. We then did actual counts of the eggs and sperm. And so the average of a number of eggs per gram of gonad is 82,000 for females and 126 million sperm per gram of gonad for males. And so we model this to see if this is related to size. And for females, the fecundity, the number of eggs, is related to the size of the starfish. But we didn't see this same relationship with males, and the asymptote was remarkably similar to previous measurements. So we looked at the fecundity. So in the extreme, large female cots bigger than 40 centimeters are estimated to produce over 100 million eggs, whereas the equivalent sized males could produce over 56 billion sperm. So this shows us that a breeding pair of mature cots could be enough to cause outbreaks. So from this to that. So this highlights the importance of detectability and establishing an early warning system for Cots outbreaks. So the question, given all this, the high fecundity, early age at maturation, I think the question would not be why outbreaks occur, but why don't outbreaks occur? So what drives these fluctuations in reproductive success? And so we looked at gametogenesis and spawning strategy in crown of thorn starfish. We did the study at in Lizard Island, and for fine-scale assessment, we repeatedly sampled this site. So this is within the outbreak initiation box, and sampled monthly between September and March. So we looked at, examined the gonads to assess their developmental and maturation stage. We looked at gonad indices and also analyzed histology of this sample. So so spawning in relatively higher latitudes like the Great Barrier Reef happens around the summer months. So in the GBR in Japan, for example, it happens around times when temperatures are approaching around 28 degrees Celsius. And these conditions are ideal for embryogenesis and larval development. So the graph on the left shows their these thresholds, and these are the 28 degrees is optimal temperature for this. We looked at the maturation stages, and we did hierarchical log linear comparisons and show that there's a difference in the relative frequencies of maturation stages between spawning seasons. So the first year, the 2013-2014 spawning season, you can see that so after spawning in December, you can still see that there's still mature and partially spawned starfish, even up to March. And the following year, after spawning in December, they've all, from January, they've all spawned. So all the gonads were either mostly spent, and some of them were partially spawned. So you can see a difference, temporal difference in the maturation stages of the gonads. And this was consistent with the Gonad index as well. And you can see the first spawning season, the one on the orange graph, you can see that it's slowly, GSI was just gradually going down. And compared to the following season, the reduction in the Gonad index was more abrupt. So for the 2014-2015 spawning season, Gonad index was relatively low at the early stages of the cycle, around September, but consistently increased in the following months until the Gonad index peaked in December, and then it drastically reduced after spawning. So this indicates complete and spawning, while the previous year it's sort of dribble spawned. We looked at the histology of this as well, and it's pretty clear. So around September to November, it's the eggs are developing. So you have mature oocytes in the lumen, and when December, right before spawning, so you have the gonads are full, and when you get to January, the 2013-2014 spawning season, you still see large and mature eggs in the lumen, but in the following spawning season, they were mostly gone. And in February as well, you still see this large eggs in the previous year. The following year, they're all gone. And so it would be good to see sample from April to August as well, to see what happens, how if gametogenesis restarts straight away after spawning. And with the males, so if we sample populations, usually after spawning, we also see like highly skewed towards males populations, because a lot of the females lose their eggs, so it would be hard to find gonads in the females, but the males can restart spermatogenesis after spawning. And this is just how so around August, September, when it's developing, the spermatogenic layer thickens, and then C there shows that it's densely packed with sperm, and then D is after spawning. So we looked at different possible environmental drivers for this spawning behavior. We looked at temperature, day length, rainfall, salinity, which were correlated, and also the chlorophyll concentration, and overlaid the gonad indices, and found that temperature and day length were correlated with these patterns. And although interannual variation of day length is negligible, so we looked at temperature. So the rise in temperature in the previous year was more gradual. So there was a 3.15 degrees Celsius temperature rise between November and December, September to December, and compared to the following year where there was a 4.43 degrees Celsius temperature rise. So this was consistent with the gonad indices and the reproductive patterns. And as you can see, the increase in 2 degrees Celsius increase between November and December in the 2014-2015 spawning season resulted in a synchronized complete spawning. So we looked at possible temperatures, a possible cue for spawning in Cots. So that we found that temperature change induced male Cots to spawn. And then once the males spawn, they have, as Scott showed, pheromones, reproductive pheromones that encourage the females to, after the males spawn, then the females follow them, but the males were more sensitive to the environmental cues, and the females were sensitive to the pheromones from the males. So in conclusion, the populations are mostly skewed towards males and have important implications on gamete release and successful fertilization. A majority of the individuals become reproductively mature around 2 years, and gradual increases in temperature up to 28 degrees Celsius, above 28 degrees, over the spawning months in 2013, 2014 caused batch dribble spawning, while abrupt increases in temperature in 2014, 2015 caused a more synchronous all-at-once spawning. So small fluctuations in reproductive output and timing, in this case in response to environmental factors, can have disproportionate effects on recruitment, and this helps us understand the spatial and temporal patchiness of Cots populations. So what are the implications of this information for Cots outbreaks? So if you model where the potential bottlenecks are, larval density or density of pre-settlement larvae, knowing the fecundity and these information on sex ratios and also reproductive behavior are important in modeling this. So high levels of fecundity point to the importance of improved surveillance of pre-outbreak populations and also establish an early warning system, for example, eDNA. Sex ratios may affect fertilization rates and also revise how these ecological thresholds are modeled. And also the temporal variability in reproduction shows that there will be problematic years where there is an anomalous increase in larval supply and recruitment. And this emphasizes the importance of detecting juveniles, as Eric showed yesterday, and also pre-outbreak, monitoring pre-outbreak populations, as well as timing culling efforts to suppress future outbreaks. And with that, I'd like to thank the following people, and thank you.